Okay, Jared, you ready to go?
Yep.
All right, let's start then. All right, so let's begin. This is Proteoform Thursdays. I'm Joe Liu from UCLA. I'll be your moderator today. And today we have the pleasure of having Professor Jared Shaw from University of Nebraska give a talk on his work with top-down mass spectrometry. But before we begin, let me just go through some introductory slides. The Consortium for Top-Down Proteomics would like to thank our major corporate sponsors, Thermo Fisher, Bruker, SciEx, and Lilly, and our consortium supporters of Agilent, Pfizer, MSC, New Objective, and Impro. Of course, this is the end of the year, and so if you need to make a tax deduction, I'm sure the consortium would be happy to take some of a donation from you before the end of the year. I'm probably not supposed to say that, but I just did. Okay, so with that, then the consortium then runs a lot of meetings and programs. Proteoforms Thursdays is just one of them. And what I would like to do is then introduce then our future Proteoform Thursday seminars. Then in January, when you come back from the holiday, then we have Professor Pei Su from a new faculty member at the University of California, Riverside, who will tell us a little bit about single molecule mass spectrometry and proteoform detection. Um, and then following then, in February, then we have an event to discuss application and challenges for top-down and native mass spectrometry in biopharma. Those will be a panel discussion from industry leaders who are interested in applying their work to pharmaceutical research. That'll be in February. And then in March, then we have Jeffrey Huang from Northwestern University will give a talk on for our Proteoform Thursday seminar. So be on the lookout then for notices then that you should receive in your inbox. All right, so with that then, let's move on then to the main event. This is a talk by Professor Jared Shaw, Department of Chemistry, University of Nebraska. Many of you, of course, know Jared, but if you don't know, I'll give it just a very brief introduction. Jared received his bachelor's degree in biochemistry from Texas Tech University, and then he later then went on to get his PhD in chemistry from University of Texas at Austin, working with Jenny Broadbell. He was a postdoctoral researcher and senior scientist at Pacific Northwest National Lab for several years. And then afterwards, then he joined the team at Emission as the chief scientific officer in 2020. He then later left the company, and then in the fall of 2023, then he moved to the Department of Chemistry at the University of Nebraska-Lincoln as a new faculty member. So with that, then I'll let Jared take over, where he'll tell us his work about ribosome heterogeneity revealed by complex up native mass spectrometry and top-down mass spectrometry or top-down proteomics. So Jared, I'll stop sharing, and you can go ahead and share your slides.
All right. Thanks, Joe. Thanks, Paul, and everybody else with the consortium for this opportunity. And so, get my slides shared. Presentation, hopefully. There we go. All right. Joe, are you seeing the
Yep, got it.
slides? Okay,
Got it. Yep.
perfect. All right. So today I'll be presenting on one project on kind of how we're starting to expand some of the top-down or utilizing top-down proteomics to kind of expand some of our native mass spectrometry workflows and using top-down proteomics to inform interpretation of complex up, so native mass spectrometry, and uh, yeah, applying this to understanding how the ribosome changes as a function of biological conditions and responses to environmental stresses, etc. So how different compositions of the ribosome enable different biological functions. And so one area or one reason why we're interested in this is that we have an emerging threat of antimicrobial resistant pathogens are an ever-growing concern to human health. And there's a number of different ways that microbes can acquire this type of resistance. And so many different mechanisms, just a few of them shown here, where microbes can block the uptake of drugs, small molecules that meant to kill the pathogens, or pump them back out of the cell, or have alternative enzymatic pathways that can go around the blocked pathway. But the ribosome is one of the major targets of many classes of antibiotics. And so if we can stop translation, we can stop production of proteins and thus cell growth, and then hopefully stop the infections, right? And so as one of the major targets of antibiotics, the ribosome, we're trying to understand now how we can find new targets within the ribosome, or understand different types of interactions within the ribosome and other ribosome-associated proteins to discover new interactions and new targets for small molecule drugs that could be utilized to fight these pathogens that are resistant to our current repertoire of antibiotics. And so the ribosome, just a little bit of general information, right? So we have 30S or 70S and 80S ribosomes, which are largely conserved across domains of life, and generally composed of about 60% RNA, rRNA, and then 40% protein, various ribosomal proteins and ribosome-associated proteins. And so that's generally 3 to 4 rRNA strands, and then between the 70S and 80S ribosome, we have 50 to 80 ribosomal proteins, and then as this slide shows, many different types of proteins, whether it's transcription or translation factors or other types of ribosome-associated proteins which modulate the function of the ribosome. And so there's a lot of different pieces that come together to regulate translation. And so what we're trying to do is come up with a fast or more efficient, cost-effective way of getting at how all these pieces come together to affect biological function. And so currently, right, we have to solve high-resolution structures with crystallography or electron microscopy, and those are long and laborious tasks and quite expensive. And so we think that native mass spectrometry and top-down proteomics offers a great opportunity to streamline and inform those high-resolution structural methods or approaches. And so as I'm alluding to, there's kind of a ribosome code, similar to what we know more commonly as the histone code. So ribosome code now in the context of translation regulation. And so how do all these different types of post-translational modifications, different compositions of subunits, ligand binding, and then all these various transiently associated ribosomal proteins come together to enable regulation of translation. And so in addition to our context of this drug discovery or targets for therapeutic development, right, there's a lot that's unknown about ribosome structure and various proteins that interact with the ribosome to affect regulation. And so just one example I'm highlighting here is this paper last year from Nature, where they discovered a new family of bacterial ribosome hybridization factors. And so this protein that's present in 20% at least of bacteria and archaea, so balin, this protein which binds to tunnel in the ribosome and halts translation, was previously unknown, but is widely utilized in many different types of or species and organisms, bacteria and archaea. But we didn't know about it because we largely focus on model organisms like E. coli and other similar model organisms. And so we think that we can use top-down proteomics in the combination with native mass spectrometry to get at what proteins are present and how it's changing and how it all comes together under different biological contexts. And so that will be the focus of today's talk. And so for those who aren't completely familiar with all of the the ins and outs of top-down proteomics and native mass spectrometry. I've just got a few introductory slides. And so I have to apologize that through various presentations, I've lost the citation for this original figure, but in the protein-centric mass spectrometry world, right, we start up with bottom-up proteomics, where we digest a protein into a bunch of pieces and we infer identity and from all the pieces, and that's analogous to all these scientists looking at a small piece of this elephant and coming up with a different identity based on which part of the elephant they're looking at. And then when we take a step back, or the bird's eye view for top-down proteomics, right, now we can say that with this more context, the zoomed out view, right, we can say this is actually an elephant. And then further, we can take a even further zoomed out view and say, like, native mass spectrometry would be then even more hierarchical organization of the components, and say that no, that's not necessarily just an elephant, it's a zoo, and this would be our native mass spectrometry type view of what's going on in biology, right? And so in this talk, we'll be focusing on these two levels of biomolecular mass spectrometry to get a better idea of what's going on in the ribosome. So we largely depend on two different types of mass spectrometry, right, where we're looking at proteins or complexes from a denaturing standpoint or native standpoint, and that largely depends on our solution conditions, right? And so with electrospray ionization, our spectra reflect the solution conditions of our analytes. And so just as an example, we have a monoclonal antibody under typical denaturing mass spectrometry conditions. And so we have many charge states, broad charge state distribution with high charge. But then if we take that same molecule and use a native mass spectrometry friendly solution, we can see we get far fewer charges and a much tighter charge state distribution, which reflects a more folded or compact gas phase structure that was present in solution. And so for a single protein, or in this case, covalently bound subunits, right, we have this kind of information from electrospray ionization. But if we move out to then non-covalent protein complexes, and this is an example from the Heck lab, where we have urease from H. pylori, right, under denaturing conditions. We have the two subunits, which appear separate in the mass spectra with their denaturing representative charge states distributions for each of the proteins. But then under native mass spectrometry conditions, right, we can observe this complex, or these proteins, as a complex. And instead of being two individual proteins, it's actually 12 alpha and 12 beta subunits together to give this megadalton complex. And so we'll be going through similar experiments like this in today's talk. So the work I'll be talking about today is how do we combine native mass spectrometry and top-down proteomics to get a better idea of what's going on in the ribosome. And so we're building on a lot of pioneering work done by Carol Robinson's lab and then Albert Heck, both in terms of dissecting the ribosome, but then doing high-resolution measurements of these intact complexes. And so what we're trying to do to expand upon that work is figure out new ways of breaking apart the intact complexes to figure out which set of the proteins and components come together to form the active ribosome that we extract from cells. And so to do this, we're going to be using top-down proteomics of some of the isolated ribosomal proteins. So we'll do, say, a glacial acetic acid precipitation of the rRNA, and then just take our denatured proteins and do a typical top-down proteomics workflow to do LC-MS/MS to identify which proteoforms of the ribosomal proteins and ribosome-associated proteins are present in that sample. And when we're talking about ribosomal proteins, we're generally talking about acetylation and methylation, but there are a number of truncations as well. And then we can get relative quantitation of these different proteoforms. But most importantly, we're going to use this data to create essentially a sample-specific database that we can use to annotate or interpret our native mass spectrometry experiments. And so what we're doing differently is now we're taking the intact ribosome and reducing the magnesium concentration, so we get the 30S and 50S subunits. So 800 kilodaltons and 1.5 megadaltons, respectively. And then we're going to take advantage of some specific activation using IRMPD, so infrared multiphoton dissociation, to hopefully, as I'll show, unfold and maybe dissociate the rRNA and eject all of the ribosomal proteins from the complex. And so the goal is to measure the intact masses of all the proteins that are coming out of the ribosome, and then combine that information with our top-down proteomics to get a better idea of everything that was in the ribosome and how they're varying as a function of different biological conditions. And so, yeah, just a little bit of information on top-down proteomics for those who are not experts. And so we do reverse phase liquid chromatography of our ribosomal proteins that we've isolated. So as a function of time, right, we've got proteins eluting from our chromatography to the mass spec. And then just as an example, I've picked this late eluting peak here at 26 minutes. And so if we look at the intact mass spectrum at that time, we get this charge state distribution of our ribosomal protein. And so from that, we can determine intact mass. And we can see that there's a couple forms of that protein with the delta mass of 14 Daltons, right? Probably corresponding to methylation. Can isolate a charge state and do tandem mass spectrometry, and then we can use the diagnostic product ions from that MSMS event to identify and hopefully localize PTMs. And so through this process, right, we can identify this chromatographic peak as protein L7, and with its product ions, then we can localize that we have acetylation at the N-terminus. And so this is our proteoform for L7. And so as it turns out, that chromatographic peak just before it is protein L12. And so the only difference between L12 and L7 is that L7 is N-terminally acetylated. So same amino acid sequence, but L7 is N-terminally acetylated. But both of those proteins can have lysine methylations at different points. And so I'm pointing out these proteins because we'll come back to this when we talk about some of the native mass spectrometry results and some of the interesting findings that we get from those experiments. And so L7 and 12 are unique among the ribosomal proteins in that they're present in 4 copies, whereas all the other ribosomal proteins are 1 copy per ribosome. And so L7 and L12 form part of the 50S ribosomal stock, which helps bring in different types of translation factors to the ribosome. And so under these biological conditions, right, this was E. coli cultured to stationary phase. And under these conditions, we see that we have a 20-to-1 ratio of L7 to L12. And that is known to vary as a function of the growth state. And so this is just a nice picture of structure of the ribosome, composite structure showing the 50S ribosomal stock, and then we have our 4 copies of L7 or L12, which kind of act as fingers or arms to bring in different proteins. And then in some previous studies by ultracentrifugation, right, we can see that as a function of growth state, right, our ratio of L7 and L12 is known to change. And so what we see in stationary phase for our reverse phase chromatography matches well with what has been previously determined by ultracentrifugation. And then very much in line with previous studies from the Robinson group, where they studied this stalk complex previously and did a lot of different studies to understand what's going on here. And so it's good that at least preliminarily we agree with previous literature. And so now to move on with the complex up part of the experiment. So complex up, right, we're taking the intact 30S and 50S subunits and then trying to break them apart and see what intact subunits come out of the complex. And so we're going to be doing that with infrared multiphoton dissociation. And then, like I mentioned, hopefully we're preferentially unfolding and activating the rRNA to kick out our intact ribosomal subunits so that we can see all the different components that are there and their relative abundances in terms of the proteoforms. So just to show that a little bit differently, right, we have our 70S ribosomes, which we isolate from cells, and then we can reduce magnesium concentration in solution. And that leads to dissociation of the 70S into 50S and 30S subunits. And then we're doing native mass spectrometry of those species, right? And so in our UHMR, we get beautiful high resolution spectra of both of those species. And as you can see, they're present in a number of different forms with the addition or loss of ribosome-associated proteins or some of the core ribosomal proteins. And so just as an example, in the 30S subunit, S1 is a protein that is known to have functions outside of the ribosome. So it's not uncommon to see 30S species without that ribosome. And then S1 also interacts with S2. And so if we lose S1, we probably can lose S2 as well. And then we observe the stock, the 50S subunit minus the stock, and then plus a silencing factor, and then as well as the intact 50S subunit, right? And so the idea is, though, although we can manage to convince ourselves that those masses correspond to those compositions, right, we'd really like to be able to break apart these species and see what's actually there. And under the factory conditions of the UHMR, we're unable to effectively dissociate these complexes with HCD alone. And so we can't at all dissociate 30S, at least in our hands, but we get a little bit of fragmentation of the 50S, but we don't really get a whole lot of information with nitrogen as the collision gas. And so this is why we're exploring infrared multiphoton dissociation. So we've taken a CO2 laser and then interfaced it with the HCD cell of our UHMR. No focusing, no collimation. So we're not trying to get the most efficient dissociation, which would probably lead to protein dissociation as well. We're trying to just heat up the rRNA, maybe fragment it, but unfold it, and then allow ejection of the ribosomal proteins. And so what makes us think that we can do that is the fact that the phosphodiester bond, the PO bonds present in RNA, DNA, right, are strong chromophores for infrared photons is 10.6 micron, whereas peptides and proteins are relatively weak chromophores. And so under our hypothesis is under appropriate experimental conditions, we can preferentially unfold and fragment the RNA while leaving the intact proteins to fall out and be detected, right? And so just real quickly to go through control to show that we were probably doing something like this, we took alpha casein, so it's a model phosphorylated protein, so about 24 kilodaltons and has 8 phosphoserine residues. And so those PO bonds, which are the chromophore for this wavelength of light. And so we isolate the 9+ charge state and try to do IRMPD, and under moderate conditions, we don't get any dissociation. But then if we compare that to a small RNA, right, so 25mer we can buy from New England Biolabs, and then under half the laser power and a small fraction of the irradiation time, we get extensive dissociation. So this leads us to believe that with the ribosome, we won't be, at least to a large extent, fragmenting proteins, but we'll be unfolding and fragmenting the rRNA in the ribosome just to kick out the ribosomal proteins. All right, so if we go back to our native MS spectra, right, so we have our 30S and 50S subunits. We're going to do quadrupole mass selection of each of these distributions and then do IRMPD with them under mild activation conditions. So 15 watts and 10 to 40 milliseconds of activation, so well below anything that we were working with for proteins and didn't see dissociation. And so if we look at that, right, so here's our full mass spectrum of that experiment where we have our precursor ions almost completely dissociated, and then abundant low MZ products, which correspond to our ribosomal proteins. And so if we zoom in on that, we've got some that come out very abundantly, and then a distribution of a lot of other ribosomal proteins. High res measurements, so we're isotopically resolved, so we can do extract deconvolution, and then get monoisotopic masses of the ribosomal proteins that we've kicked out. And so this is partially annotated spectrum, but we can see, like for S6, we see various forms, and then also similar for S2, where we can see methylated forms, etc., right? And we can do the same for the 1.5 megadalton 50S ribosome subunit. And so similarly, extensive dissociation of the precursor ions and a nice distribution of ribosomal proteins, ribosome-associated proteins are kicked out. And so similarly, deconvolution shows us there's a lot of different forms of different ribosomal proteins with methylation, acetylation. We can even see some with zinc or magnesium bound, and then some ribosome-associated proteins. So this RSFS, which is a silencing factor. So we were really excited to see this because our hypothesis, at least first glance, is holding true, and we're getting a nice distribution of ribosomal proteins and extensive dissociation of both subunits. And so just to summarize what we saw there, right? So we can do this experiment both in positive mode and in negative mode. But the ribosomal proteins, right, they're interacting with rRNA, so a lot of negative charges on the RNA. So the ribosomal proteins have a lot of positive charges, so or basic amino acids. So it's not surprising that we see most of the ribosomal proteins in positive mode. So 30S and 50S, and then our ribosome-associated protein. And so altogether, we're able to see about 60% of all of the ribosomal proteins from these two different experiments, right, on the 50S and the 30S subunits. And so the next thing we want to do is try to start justifying why we see and what we see and the abundances. So we found a couple structures of the free 30S and 50S subunits, and what we color-coded here in red are the ribosomal proteins that we see in our complex up analysis. And so what I'd like you to just take away is that there's not any certain locations that we're seeing the ribosomes exclusively from. They're more or less evenly distributed around the ribosomal subunits. And yeah, so we wanted to then dig a little bit deeper and trying to justify the abundances that we see and whether and why we don't see certain ribosomal proteins. And so we calculated the surface areas and the interface areas for, this is just using the 50S as an example. So we have our proteins, 50S ribosomal proteins that were present in the structures, and then interface area, total surface area. And so we found that it correlates pretty well with our relative interface area. So for example, protein L9, 5% of its surface area is involved in an interface within the ribosomal, the 50S subunit. And so that is here, right? So we have this large or long elongated structure of L9 and this very small interface patch with the rRNA, the 23S rRNA. And so naturally, if we're giving these ions vibrational energy through IR photons and unfolding the ribosomal proteins and RNA, we would expect that L9 was very abundant and would fall off easily, and that's exactly what we see. And so the shaded columns in this table are the ones that we observed in our IRMPD analysis of the 50S subunit. And so you can see, we see most of the proteins that are in these low percentage of interface area, but we do see L20, which has the highest interface area, which is a bit interesting. And so it's right here. And so it interacts with a couple of other ribosomal proteins and then is in a fold of the 23S rRNA. And so naturally, as we start kicking out the ribosomal proteins that it's associated with or interacting with, and as we unfold maybe the surface of the 23S RNA, its actual interface area probably drops quite significantly. And so maybe that's a good justification for why we see L20 relative to some of the others. Well, whereas L34 is buried really deep inside the 23S RNA. We're unlikely to ever see that because we just have to do so much unfolding of the large megadalton rRNA strand. And so we wanted to then characterize, see if we can do these same types of experiments as a function of cell culture state. And so we grew E. coli under a number of conditions, right? Log phase, late log phase, and stationary phase. And then we extracted the ribosomes and did the same kind of procedure where we reduced the magnesium and then do native MS of the 30S and 50S subunits. And so I won't go into a lot of the details here, but we can see there's definitely trends and changes and association of a number of different ribosome-associated proteins as we get into stationary phase, known proteins that are used to regulate translation or inactivate the ribosomes, so RA, IA, and then similarly with our 50S subunit, right? And so then we went through and did IRMPD of the 30S and 50S subunits in all of these conditions. And so what I want to point out is kind of how well do our top-down and native mass spectrometry workflows agree in terms of proteoform abundances. And so just to show initially that we're seeing things that make sense from our IRMPD experiments, right? So we're pulling out kind of distributions of protein S6 and then L7 and 12, as we talked about earlier. And so S6 is known to be C-terminally glutamylated, and then the extent of C-terminal glutamylation has a distinct shift once the cells go into stationary phase. And so we can see that clearly where we have unmodified S6 and then completely modified and then varying amounts of C-terminal glutamylation in S6. And then as we would expect based on the previous literature, right, under log phase, we have greater contributions of L12 to the stop complex. And then as we move into stationary phase, L12 is almost completely replaced by L7. And so this is good agreement with previous literature. And so I think our methods are reliable and giving us some useful information as we move forward in different studies. And so how well do our top-down proteomics and complex-up experiments agree in terms of these relative proteoform abundances as we look at the ribosome under different culture states? So just remind you, so we have log phase, late log phase, and then stationary phase. And so we're looking at here the intensity weighted number of C-terminal glutamylation on S6. And so you can see we have really good agreement between IRMPD and top-down proteomics for all of these. A small but maybe statistically different number in stationary phase, but largely in agreement. And so we can say that we don't expect the C-terminal glutamylation to modulate interactions of S6 with the rest of the ribosome because it's at the C-terminus, and the C-terminus is known through structures to not be interacting with any other parts of the ribosome. But as we move into L7 and L12 and comparing the ratios we see there, where we have in stationary, log phase and late log phase, and then stationary phase, you see in log phases, we're very good agreement between IRMPD and top-down proteomics. But as we move into these high L7 compositions of the stop complex, right, it's known that incorporation of L7 makes the stop complex much more stable and more tightly bound to L10, or L11, which binds it to the ribosome, to the 50S subunit. We see this dramatic underestimation, I guess you would say, by IRMPD of the amount of L7 in the ribosome, right? And so we think we're stumbling into a different method or new method for getting at proteoform-specific relative interaction strengths within the ribosome. And so, right, we're underestimating the amount of L7 due to stronger interactions, so able to completely dissociate the L7 from the ribosome, whereas top-down proteomics, right, denaturing conditions, liquid chromatography is giving us the true answer in terms of the L7 to L12 ratio. And so This is an active area of research, and we're trying to find additional examples for this. But given the previous work by the Robinson lab, we're feeling pretty good about these at least initial observations. And so with that, I can conclude that hopefully I've shown you we've developed a pretty useful approach where we combine top-down proteomics with complex up native mass spectrometry to get at how ribosomes are modulated, or their structure and composition is modulated. And then we're using IRMPD to efficiently dissociate the subunits to kick out these ribosomal proteins, which now enables us to measure these different proteoform compositions as a function of biological state. And then we're pretty excited about what might behold in these upcoming experiments for getting more at some of the biophysical aspects of the proteoforms. So with that, I'd like to acknowledge great grad students I have. Sachin Tinakum did a lot of this work. He was my first graduate student, and I had a couple of really good NSF-RU interns, so undergrads working in the lab over a couple summers, getting us going in cell culture and a lot of these more biological type experiments. And then collaborators at Thermo, Wei Jing and Rosa, really helping me get started and get some very nice instrumentation into my lab to be able to do these experiments. And then recently been fortunate enough to get some funding from the NIH to support all this work. And with that, I'd be happy to take any questions.
Okay, thanks, Jared. That's a super interesting talk, combining top-down mass spectrometry and native mass spectrometry to look at the structure of the ribosome. I guess as moderator, I'll take the prerogative to ask the first question. So just to be clear then, that your top-down proteomics experiment, you see all of the subunits, or you can catalog all the subunits by that experiment, but you don't see every subunit in your complex up experiment. Is that correct?
Correct. Yeah. So the only ribosomal protein we don't see in top-down proteomics is S1, and it's 60 kilodaltons, right? So
Yeah.
just not favorable for top-down, but we do
Yeah.
see it by bottom-up. So yeah, they're all
So
there.
then when you blast it with the IR laser, do you vary the laser power or the fluence to get some additional information about the interactions of the ribosome of the proteins to the RNA?
Yeah, absolutely. So that's something that's expanding on some of those later aspects I was talking about in terms of what do we see and why do we see it and the abundances. And so yeah, it's tricky. We're still developing all of our refining our methods for getting stable spray for the ribosomal protein or subunits under over a long period of time. Uh, and so that's literally the limiting factor between doing all these different variable power and experiments. But we have done that, and we do see a nice increase in like the most labile, you would say,
Mhm.
interactions. And then as we irradiate longer, we do see other proteins grow in. And so yeah, there's definitely information to be had there. We're still working on it.
Yeah, that's neat. Okay, so let me read off some questions. Also, so if you have questions, you can type them into the chat, or you can raise your hand and hopefully I'll see your hand. So let's start off with some of the typed questions. This is from Mike Senko. Do the proteins you see coming off the ribosome correlate, he thinks inversely, to molecular weight?
Yeah, well, so of the ribosomal proteins, a vast majority of them are between, about 9 kilodaltons and 15 kilodaltons. So in terms of charge state dilution, isotope dilution, we wouldn't expect there to be a huge difference there. But for some of the larger proteins, like they're in the 20 to 20 kilodalton range, we definitely see their lower abundance.
Yeah,
Tough to back out whether it's due to those factors or being more tightly bound to the ribosome. So it's a bit convoluted there. Yeah.
yeah, yeah. David Butcher then asks, if the interface area is separated into protein-protein and protein-RNA interface
Mm.
areas, is the same trend observed with proteins with lower relative interface areas dissociating more readily?
No, that's an excellent question. We, as far as I know, we haven't looked at that specifically yet. It's just been we tabulated the total interactions, both protein and RNA. but that's something we should definitely look into. Yeah.
I'm assuming you could look at other RNA, sorry, other ribosomes from other species then that give you some slightly different compositions, slightly different RNA structures.
Yeah, absolutely.
Yeah,
Yeah.
yeah. All right, so Paul Danis then he asks, could you explain a little bit more about knowing the composition of the ribosomal proteins, how they will impact the battle of drug resistance?
Right, right, right. So I guess understanding which subunits are there, what forms of those subunits are changing as a function of biological conditions, or which ribosome associated proteins are then interacting with the ribosome under different conditions, right? Understanding those interactions or finding those proteins that are there and we just don't know about, right? Gives us new targets or new interactions that we can design small molecules for. And I think in that introductory slide, I had valin, so that new protein that was discovered, right, that
Yeah.
blocks the whole tunnel through the ribosome, right? And so if you could design a small molecule that locks that in place and halts translation, right, then that's a pretty good way for killing a microbe, right?
Yeah, is balin in the E. coli ribosome?
It's not. Yeah, that's why they say
Oh, okay.
we're too much emphasis on model organisms, and so yeah, we're gearing up to grow a bunch of different types of things and see what we find.
Yeah, cool. All right, Nick Young, he says, Jared, great talk. Throughout, I was wondering about how ribosome composition relates to subcellular localization. In eukaryotes, there's emerging evidence of differential ribosomal composition depending on location. In
Yeah,
bacteria,
absolutely.
there is different subcellular localizations depending on growth phase. So do you see, or are you tackling that question? That's a
Yeah,
long
that's another layer to of difficulty, right? So, absolutely, yeah, that's one of the things that we want to go after, right? And yeah, bacteria or other prokaryotes, right? And then we could have cytosolic ribosomes versus membrane-bound ribosomes, and
one.
how is that interaction modulated, and do the compositions vary there as well? But yeah, we definitely plan to move into eukaryotes very soon, and
Yeah,
and
yeah.
maybe doing organelle isolation and everything else. So mitochondria or Golgi, et cetera, right? And see what subpopulations of ribosomes are associated with those different
hmm
locations. Yeah.
hmm Um, oh, okay. Lloyd Smith has a question now. Have you or anyone else thought much about how post-translational modifications in ribosomal RNA might also have a code that affects
Absolutely.
biology, perhaps interacting with the protein code?
Yeah, there has to be. There definitely has to be. I think it's maybe not as well documented in the context of the ribosome, but yeah, definitely post-transcriptional modifications play a huge
Right.
role in ribosomes or RNA structure and interactions. And uh, I think there's just not a huge amount of literature and evidence for it yet, specifically for the ribosome. But yeah, like Dan Fabrice at UConn, it's exactly what he's focusing on and trying to, yeah, the RNA modification code, I guess you could say. Yeah.
Yeah, I mean, Nick says the same thing, RNA modification. So in that vein, then, when you turn on the IR laser, then
Mhm.
you're using that to get rid of the RNA, but what happens to the pieces then? Do you
Yeah,
do you
exactly.
do you see RNA pieces?
So largely, no. There are less than 2,000 MZ. We do see some things we can't explain. So it's
Hmm.
probably RNA fragments. In negative mode, there are a lot more unexplained fragments and pieces, and so presumably we would assume that those are RNA product ions. Uh,
Yeah.
we do currently we focus on high res and low MZ stuff, but we have done a few experiments trying to look at the remainder part that we in the high MZ that we uh, the results from the proteins falling out. We just don't have the resolution or enough homogeneity in those fragments to really do any good assignments right now because they're large and right these large RNAs could fragment in many different ways and yeah,
What if you were
we're thinking
to,
about it. Yeah.
yeah, so to get rid of the RNA, there are other ways to do that. Couldn't you just chuck in a nuclease and just digest the RNA out? And would the proteins still associate together when you do it that way?
Hmm. So have pieces of the rRNA with proteins bound and
Yeah,
yeah.
yeah. I don't know if that would
I,
be better than the laser. I don't know. I don't
I'm
know.
not sure. I think something could be figured out, but yeah, a lot of the surface area of the RNA is protected by the proteins, and I'd have
Yeah.
to think about that more. Yeah.
So there was another question, sort of in that vein. Have you considered whether a combination of activation methods is necessary? So a combination of activation methods, or if just IRMPD alone is sufficient?
Yeah, absolutely. So we've been talking with the Wysocki lab, and so we're
Yes.
developed a new SID cell that'll go in a different position in the UHMR and hopefully give us more and complementary information to the IRMPD. So yeah, that's definitely underway, and hopefully we'll have some of those first experiments done in the next couple weeks.
Mm-hmm. But I mean, you have the IR laser going through the HCD cell. So do you turn on HCD also when you do this experiment?
With how we're currently triggering the laser, we use one of the voltages on the HCD cell, so we can't currently do the combination of HCD and IRMPD. It's either or. But yeah, we've figured that out as well, and those are also experiments to come.
Okay. All right. Yeah, that's cool. All right. Other comments like, great work, Jared. That's from Lily. Brune has says the same thing. So does anybody else have any questions? We're almost out of time. Okay, one more. Oh, no, that was just great talk. You can see the comments, Jared. All right, then I think we'll close today. That was a great talk, Jared, and that was a great end of our Thursday Proteoform session. So thanks everybody for attending today's meeting. If you're interested in microbial or antimicrobial resistance, as president of ASMS, I can remind you that at the end of January, there is a conference hosted by ASMS in Santa Fe on mass spectrometry in microbial science. And so that will be a topic, I think, of that conference. So if you're interested in hearing more about this particular topic, not native top-down mass spectrometry, but antimicrobial resistance, that would be a good meeting to attend. So with that, thanks a lot, Jared, for contributing to our session. And we'll close today. Happy New Year, happy holidays to everybody, and we'll see you when the new year starts.
All right.
Thanks very much.
Thanks, Joe. Thanks, Paul.